I'm Rhena Schumann. I'm from the University of Rostock and I'm the head of the Biological Station in Zingst. I studied marine biology many years ago. The biological station is situated at the southern Baltic in a so-called inner coastal water, which is a lagoon of brackish water, and it's heavily eutrophicated, and therefore in the 60s there was a program of monitoring installed by the state agency together with the University of Rostock, and we started with simple hydrology and then added parameter by parameter like water chemistry, nutrients, and later organisms like phytoplankton, zooplankton, bacteria, and a lot of colleagues worked on benthic organisms, but that program is discontinued so far. So we started with data collection in the 60s, and we have many data on biodiversity of plankton organisms, and it's not so easy to publish data if you have to provide the data. And as you can imagine, biodiversity data are not simple parameters with one value, but you have a lot of organisms in one sample, and so the data gets more and more complex. Therefore, we have to find a deposit for the data, or repository to provide data for others to use, but also for us ourselves to get some more tools, or to get help or cooperation with other scientists to interpret this data on the long course. There is the assumption that, for instance, cyanobacteria, blue-green algae, are bad for the ecosystem, for ourselves, because they can produce toxins and they can fix nitrogen to overcome some nutrient limitations. So they are self-sustaining. If they just won the system over, then they can continue on their own. And the question is that true? And we found out 10 years ago that our cyanobacteria don't do that. They cannot produce toxins. They cannot fix nitrogen, most likely not, because they don't have the genes for that. But are they bad in themselves anyway? So, and then we found out that these special cyanobacteria have a very bad impact on underwater climate. So if they once sit there, they can be there for a very, very long time. And it's not expected that the system recovers on its own so fast than expected from other systems. So that would help to correct the expectations of restoration measures, for instance. Such monitoring is a value in itself, to see what happens and to learn for other systems, because we do not have the only of these systems, but they are sitting at the southern Baltic coast one by one. So they are very common, and even worldwide, lagoons are very common ecosystems. I'm a so-called data provider, I learned today. So I have to take the samples, process the samples, and we are not so many persons doing that. But we have now the data, and so what? So we have to make some use of that and to provide them to others. And what I expect is to learn how to do that. I did not know about this digital twin, but the biological station is part of the LTER consortium. This is long-term ecological research, and they talk a lot about data repositories. but only on a very global way, so that they are and you have to provide. And then there is still the gap I described already, how to do that. And as far as I understand, these data repositories, they can harvest each other. So if I provide into one system, I hope that other systems who need the data can just easily get them out of here.